Protein synthesis inhibitors include many different classes of medications that prevent bacterial ribosomes from synthesizing proteins. The ones that target the 50S subunit of the ribosome include chloramphenicol, macrolides, lincosamides, and oxazolidinones. Okay, first let's look at how genes become proteins. There's two steps, transcription and translation. During transcription, a specific gene on the DNA is read and a copy is made called a messenger RNA, or mRNA, which is like a blueprint with instructions on what protein to build. Translation is also known as protein synthesis, and that's when organelles called ribosomes assemble the protein from amino acids within the cytoplasm. Now, prokaryotic cells, like bacteria, have smaller ribosomes than eukaryotic cells, like those found in humans. Bacterial ribosomes are made up of a 50S subunit and a 30S subunit, which combine to form a 70S ribosome. Eukaryotic ribosomes are made up of a 60S and a 40S subunit that form an 80S ribosome. Since these proteins are different, we can create medications that selectively interfere with just the bacterial ones. Protein synthesis involves initiation, elongation, and termination. In bacteria, initiation occurs when the 50S and the 30S subunits bind to the mRNA sequence to form a ribosome mRNA complex, also called the initiation complex. The mRNA serves as a blueprint for the protein that will be synthesized. It's made up of 3 nucleotide-long sequences called codons, on top of which transport RNA, or tRNA, carrying amino acids can bind with their matching anticodon. The complete ribosome has 3 sites where tRNA can enter and bind. These are called the A, or aminoacyl site, the P, or peptidyl site, and the E, or exit site. Elongation starts when the first tRNA, carrying a formal methionine amino acid, enters the P site and binds to the start codon. This causes a conformational change which unlocks the A site for the next tRNA. That next tRNA binds at the A site. The amino acid detaches from the tRNA in the P site, and a peptide bond is formed by an enzyme called peptidyl transferase between the amino acids in the P and A sites, a process known as transpeptidation. Now, the A site has the newly formed peptide chain dangling from it, while the P site has an empty tRNA with no amino acids. In the final stage of elongation, the tRNA in the P site slides over to the E site, and the tRNA in the A site slides over to the P site. The ribosome slides over, so the free A site is over the next codon on the mRNA in a process called translocation. Next, a new tRNA with the matching anticodon binds, and the process repeats until a long peptide chain, called a protein, is synthesized. Okay, so let's look at some protein synthesis inhibitors. Chloramphenicol is a bit of a loner and is the single representative in its group. It works during elongation by inhibiting peptidyl transferase, thus preventing peptide bond formation between new amino acids and the polypeptide chain. It's a broad-spectrum bacteriostatic medication, meaning it limits the growth of bacteria rather than eradicating them. It's usually administered parenterally, and it readily crosses the blood-brain barrier, so it could be used to treat bacterial meningitis. Resistance to chloramphenicol occurs through acetyltransferase enzymes, which add an acetyl group to chloramphenicol to inactivate it. Chloramphenicol is effective against some strains of Haemophilus influenzae, Neisseria meningitidis, Bacteroides species, and Rickettsia species. However, due to its severe toxicity, it's no longer used systemically in most countries with access to safer alternatives. It is also commonly used in the form of eye ointments to treat bacterial conjunctivitis. For the side effects, it's very toxic to the bone marrow and could kill off the hematopoietic cells, leading to bone marrow suppression. This will usually lead to aplastic anemia first, followed by a decrease in platelets and leukocytes. It's also notoriously teratogenic, as it readily crosses the placenta, so it should not be given to pregnant people or newborns. This is because infants lack the hepatic enzyme glucuronidase transferase, which normally metabolizes chloramphenicol. When this medication accumulates, it causes gray baby syndrome, where the infant is anemic and cyanotic, where the skin is pale or a grayish color, and it can lead to cardiovascular collapse. Chloramphenicol also inhibits the hepatic enzymes in the cytochrome P450 family. These enzymes break down many other drugs, like warfarin. It increases the action of those medications. Next, let's look at the macrolide antibiotics, which include erythromycin, azithromycin, and clarithromycin. There's also a novel macrolide called fidaxomicin. They work by binding to the 50S subunit of the ribosome and blocking translocation, so the ribosome can't slide to the next codon on the mRNA. Most macrolides can be administered perorally as well as parenterally. Fidaxomicin is administered only perorally, and it's not absorbed in the systemic circulation. It is only used for Clostridioides difficile infection that causes pseudomembranous colitis. Now, in general, the rest of the macrolides are broad-spectrum bacteriostatic medications effective against gram-positive and gram-negative bacteria alike. Specifically, they are used as the first-line therapy for Bordetella pertussis, which causes whooping cough. They can also be used to treat typical pneumonia as well as atypical pneumonia caused by Mycoplasma pneumoniae or Legionella species bacteria. Clarithromycin is used in combination with amoxicillin and omeprazole to treat Helicobacter pylori, which causes peptic ulcers. Azithromycin is often used to treat sexually transmitted infections like chlamydia. Finally, resistance against macrolides typically involves developing efflux proteins that actively pump macrolides out of the cell. In addition, methylase enzymes can alter the ribosomal target site of the drug, and thus decrease the drug binding. Side effects of macrolides are rare, and the most common is gastrointestinal problems like diarrhea, nausea, and vomiting. The more serious side effects include a prolonged QT interval, so they should be avoided in people with arrhythmias, and hepatotoxicity, so they're contraindicated in people with liver disease. Both erythromycin and clarithromycin can inhibit cytochrome P450. Lincosamides are represented by clindamycin, which is the most commonly used member of the family. They function similarly to macrolides by binding to the 50S subunit of the ribosome, and they inhibit translocation. Clindamycin can be given perorally, parenterally, or as a topical cream. It's used to treat anaerobic bacterial infections of the lungs and mouth, in other words, above the diaphragm. It is also used for gram-positive bacteria, such as group A streptococcus, especially in patients with penicillin allergy. Lincosamides are also used in toxin-mediated conditions, such as toxic shock syndrome, because they can reduce toxin production by inhibiting protein synthesis. Topical formulations of clindamycin can also be given to treat acne. Clindamycin tastes extremely bitter, so it's not commonly prescribed to children. Common side effects include GI distress like diarrhea, nausea, vomiting, and cramps. Long-term use can cause bacterial superinfection by Clostridioides difficile, formerly known as Clostridium difficile, which can cause pseudomembranous colitis. Oxazolidinones like linezolid are a new group of drugs. Their method of protein synthesis inhibition involves binding to the 50S subunit of the ribosome and preventing it from combining with the 30S subunit to form the initiation complex. Linezolid therefore stops protein synthesis before it even begins. It also can be used parenterally as well as perorally. The oxazolidinones have a unique structure and mechanism, so few organisms have developed resistance to it yet. Those that have typically have mutations in their ribosomal RNA that prevents the medication from binding. Now, linezolid is effective against a wide variety of gram-positive bacteria, but it's often reserved for bacterial infections that are resistant to other antibiotics. This includes methicillin-resistant Staphylococcus aureus, or MRSA, and vancomycin-resistant Enterococcus faecium, or VRE. When used for short periods, linezolid is a safe drug with minor side effects like diarrhea, nausea, vomiting, and rash. But if used over a long period of time, patients begin to show signs of bone marrow suppression, which often manifests as thrombocytopenia. Another problem is neurotoxicity, and linezolid can cause peripheral neuropathy and optic neuropathy, characterized by a washed-out perception of colors as well as progressive loss of vision. Another interesting effect is that linezolid is also a weak inhibitor of monoamine oxidase, an enzyme inside neurons that breaks down the neurotransmitter serotonin. So if a person is also taking antidepressants that increase serotonin, like a monoamine oxidase inhibitor, MAOI, or a selective serotonin reuptake inhibitor, SSRI, they could develop serotonin syndrome, which is characterized by flushing, extremely high body temperature, muscle rigidity, and a tremor that may progress to seizures. Alright, now let's make a mnemonic to help you memorize and retain all of these pharmacology facts. So let's set the scene by going to an apartment where a lady is celebrating her 50th birthday. This will help you remember all of these drugs act on the 50S ribosome. Now, she's putting the candles onto the cake, which represents the formation of peptide bonds by peptidyltransferase. The drug that inhibits this step is chloramphenicol, which will be represented by a large coral growing out of the cake. Let's also put some red eyes all over the coral to help you remember that it's used topically to treat bacterial conjunctivitis. Now, to memorize the harmful side effects, let's put some broken bones sticking out of the cake for bone marrow suppression. Next to the cake are some red plastic plates that look like red blood cells, which represent aplastic anemia. So you remember this medication is contraindicated for infants, and during pregnancy, the lady is pregnant and carries a gray-colored baby doll to help you remember gray baby syndrome. A cell that's chrome-colored, representing cytochrome P450, ate some of the coral and died, which will help you remember chloramphenicol is a cytochrome P450 inhibitor. Okay, moving on. Translocation will be represented by the door into the apartment, and two groups of drugs will inhibit this process. On one side of the door, we have 4 mice carrying large lids for the macrolides. One of them is red for erythromycin. One is wearing an Aztec headdress for azithromycin. One is transparent or clear for clarithromycin. And one is wearing a fedora and holding an axe for fidaxomycin. For indications, let's put a coughing whooping crane behind the mice to represent whooping cough. Next to it, there's a very sick person coughing in bed for typical pneumonia, and another person with a walking stick who represents atypical or walking pneumonia. You can see a slice of pie in the clear mouse's stomach for H. pylori, and the Aztec mouse is standing on a large clam for chlamydia and other STIs. The intestines of the mouse with the fedora and axe are showing for Clostridioides difficile infection that causes pseudomembranous colitis. For side effects, a long-tailed squirrel who's quite a cutie wants to join the mice squad, and this represents long QT syndrome. It's a bit sick, however, and is vomiting, which represents GI distress and a bad first impression on the mice it wants to impress. Okay, let's move on to clindamycin, which also blocks translocation. So let's put a mouse dressed as Cinderella on the other side of the door. Now, instead of a pumpkin carriage, let's have her inside a fishbowl, which represents an anaerobic environment. The fishbowl is sitting on top of a diaphragm. To help you remember, clindamycin is used to treat anaerobic infections above the diaphragm. The Cinderella mouse is worried about her acne, which is another indication for this medication. For side effects, this medication causes GI distress. So, let's say the stress is giving our poor mouse stomach pains and she's vomiting outside the fishbowl. The vomit goes into the closet laying flat on the ground, which will help you remember long-term use of clindamycin can lead to superinfection of Clostridioides difficile. Okay, the last medication is linezolid, and we'll use a pan lid with black and white lines on it. This drug inhibits the formation of the initiation complex where the 30S and 50S subunits join together along with the mRNA. So, we can have a pair of newlyweds joining the party. They can represent the 50S and 30S subunits. The pan lid is the gift they bought, and they're carrying it between them, which keeps them separated. For indications, let's have the son of the couple playing with a remote-controlled Mars rover for MRSA. He's using a VR headset to control it, which stands for VRE. And now for those side effects. The kid's other toy is a dead knight who's about to be cremated, and he represents serotonin syndrome. His body is stiff, so there's muscle rigidity, and the hot fire represents hyperthermia, and the armor is glowing red for flushing. Let's wrap his arms in bandages to represent peripheral neuropathy, and instead of a sword, he's holding a broken bone for bone marrow suppression. Alright, as a quick recap. There are numerous classes of antibiotics that inhibit protein synthesis. Many of these act on the 50S subunit of the ribosome, but their mechanisms can be very different. Oxazolidinones, like linezolid, stop the initiation complex from forming. Both the macrolides and lincosamides, like clindamycin, prevent translocation. Chloramphenicol inhibits peptidyltransferase, which is the enzyme that creates the peptide bonds. But wait, there's more. Here's a mind map with all of the mnemonics from the video. Go ahead and pause so you can test yourself to see what you remember. Stay tuned for the answers after the credits.
Aminoglycosides are antimicrobial antibiotics that inhibit bacterial ribosomes, which are the organelles that make proteins. Genes are used to synthesize proteins in two steps: transcription and translation. During transcription, a specific gene on the DNA is read, and a copy is made called a messenger RNA, or mRNA. Translation is also known as protein synthesis, and it's when ribosomes use mRNA to assemble proteins from amino acids within the cytoplasm. Now, prokaryotic cells, like bacteria, have smaller ribosomes than eukaryotic cells, like those found in humans. Bacterial ribosomes are made up of a 50S subunit and a 30S subunit, which combine to form a 70S ribosome. Eukaryotic ribosomes are made of a 60S and 40S subunit that form an 80S ribosome. Since these proteins are different, we can create medications that selectively interfere with the bacterial ones. In both eukaryotic and prokaryotic cells, protein synthesis involves initiation, elongation, and termination. In bacteria, initiation happens when the 50S and 30S subunits bind to the mRNA sequence to form a ribosome mRNA complex, also known as initiation complex. The mRNA serves as a blueprint for the protein that'll be synthesized. It's made up of 3 nucleotide long sequences, called codons. Transport RNA, or tRNA, carrying different amino acids can bind to these codons with their matching anticodons. The complete ribosome mRNA complex has 3 sites where the tRNA can enter and bind. These are called the A, or amino acyl site, the P, or peptidyl site, and the E, or exit site. Elongation starts when the first tRNA, carrying a formal methionine amino acid, enters the P site and binds to the start codon. This causes a conformational change in the ribosome, which unlocks the A site for the next tRNA. A process called proofreading happens here, where only tRNAs with the matching anticodon can bind to the corresponding mRNA codon. After the next tRNA binds at the A site, the amino acid detaches from the tRNA in the P site and gets attached to the amino acid in the A site by the enzyme peptidyltransferase. This step is called transpeptidation, because the peptide chain is transferred from the P site tRNA to the A site tRNA. Now the A site has the newly formed peptide chain dangling from it, while the P site has an empty tRNA with no amino acids. In the final stage of elongation, the ribosomes slide across the mRNA, and the A site sits above a new codon. The tRNA that was in the A site slides over to the P site, and the tRNA in the P site slides over to the E site. Now a new tRNA with a matching anticodon binds, and the process repeats until a long peptide chain, called a protein, is synthesized. Finally, termination happens when the ribosome comes across a termination codon on the mRNA. There are no tRNA anticodons that can bind to these, so they signal the end of protein synthesis. To start working, aminoglycosides need to enter the bacteria, and frankly, they aren't very good at it. Gram-positive bacteria have a thicker wall compared to gram-negative bacteria, so aminoglycosides can't penetrate them. They also require an oxygen-dependent co-transporter on the cell membrane to be transported into the cell. Obviously, these aren't seen in strictly anaerobic bacteria, so we can rule out aminoglycosides as a treatment for anaerobic infections. So on their own, aminoglycosides can only target gram-negative aerobic bacteria. Once they enter the bacteria, their main mechanism of action is to irreversibly bind to the 30S subunits and prevent the formation of the ribosome mRNA complex, thus inhibiting the initiation of protein synthesis and reducing the amount of proteins being synthesized. They can also interfere with the proofreading process, thus causing errors in the protein's amino acid sequence. These faulty proteins will eventually lead to the death of the bacteria. Finally, a 30S subunit bound to an aminoglycoside can get stuck to the mRNA. This prevents translocation, where the ribosome slides over to the next codon. Depending on the concentration applied, aminoglycosides can work as bacteriostatic, which stops the bacteria from multiplying, or bactericidal, which outright kills them. Aminoglycosides are also known for their strong post-antibiotic effect, because they can remain effective hours after their levels have dropped below the minimum inhibitory concentration, or MIC, which is the minimum concentration that inhibits the growth of a microorganism. Examples of the aminoglycoside family include amikacin, gentamicin, neomycin, streptomycin, and tobramycin. They're used to treat a wide variety of infections in the respiratory tract, urinary tract, blood, bone, and soft tissues. It's used mainly against aerobic gram-negative bacteria like Proteus species, Escherichia coli, Klebsiella pneumoniae, Enterobacter species, and Serratia species. Streptomycin is often used against Mycobacterium tuberculosis, which thrives in the oxygen-rich lung tissue. Tobramycin is given topically to treat eye infections, or in a nebulized form to treat Pseudomonas aeruginosa infections in people with cystic fibrosis. Now, remember how the thick cell wall on gram-positive bacteria stops aminoglycosides? If we add a cell wall synthesis inhibitor, like beta-lactam antibiotic like penicillin or vancomycin, we can effectively demolish or weaken the cell wall, allowing aminoglycosides to slip through. A combination like this is used to treat gram-positive enterococci infections like infective endocarditis. Aminoglycosides are not absorbed from the gut, so neomycin is often given before colorectal surgeries to eliminate bowel flora and reduce chances of infection after surgery. For systemic infections, they're given via the intravenous or intramuscular route. They can also be used topically in the form of creams, gels, or drops. Bacterial resistance to the aminoglycosides can be seen in certain strains of Pseudomonas aeruginosa, Staphylococcus aureus, and Enterobacter species. These bacteria develop transferase enzymes that modify aminoglycosides through O-phosphorylation, O-adenylation, or N-acetylation, which inactivates the aminoglycosides. Another important mechanism of bacterial resistance to aminoglycosides are mutations of the genes that encode for bacterial ribosomal proteins, leading to altered ribosome structure that aminoglycosides cannot bind to. Aminoglycosides are ototoxic and can damage the cochlea and hair cells in the ear. This effect is enhanced if the person is taking loop diuretics, which are also ototoxic. Another side effect is nephrotoxicity, which can lead to kidney failure. Aminoglycosides inhibit the release of acetylcholine from motor neurons at the neuromuscular junction and cause weakness or paralysis. At high doses, they can even cause potentially fatal respiratory paralysis. This is why they're contraindicated in patients who suffer from myasthenia gravis, a neuromuscular disorder where antibodies bind to and inhibit the acetylcholine receptors on muscle cells. Other nonspecific side effects include nausea, vomiting, and allergic reactions. Aminoglycosides also have teratogenic effects, like deafness, and thus they're contraindicated in pregnancy. Alright, so let's make a mnemonic to help you memorize and retain all these farm facts. All the aminoglycosides are having a wild party. First, we have the medications with mycin in their name. There's a mouse with wild stripes painted on his body for streptomycin, a mouse with crazy neon glow sticks for neomycin, a particularly brave mouse is riding a small cobra for tobramycin, and a mouse dressed as a dapper gentleman for gentamicin. Amikacin is not a mouse, but a human stinger holding a giant mic. For the common gram-negative bugs treated by these medications, we have an angry neighbor holding a protest sign for Proteus species. She also brought her friend, who's carrying a large club for Klebsiella, and her border collie for E. coli. Now, since sausages are made of intestine, let's use one to represent Enterobacter. The border collie is cutting it into smaller pieces with a serrated saw, which stands for serratia. The cobra has someone's eye in its mouth. To help you remember, tobramycin is used to treat eye infections. The eye came from the Mona Lisa, who has cysts and scars on her face to represent Pseudomonas in cystic fibrosis patients. Next, we have the striped mouse, or streptomycin, getting his head stuck in a toilet paper tube for tuberculosis. Finally, we have neomycin, the mouse with the neon glow sticks, who's actually a surgeon. At the party, he's operating on a patient's colon while using the glow sticks for illumination. Now, someone used a giant pen as a battering ram and smashed a hole in the purple wall. This is because we use beta-lactams, like penicillin, to break down the cell wall of gram-positive bacteria, which stain purple. This is nice because now the aminoglycoside mice can go through the wall whenever they want. For side effects, some of the mice have been nibbling on a large ear and a kidney, which represent ototoxicity and nephrotoxicity. One mouse refused to eat, so he's too weak from the hunger to move. Another mouse with a red rash was so grossed out, he started vomiting. Alright, as a quick recap. Aminoglycosides are protein synthesis inhibitors that bind to the 30S ribosomal subunit. Their main mechanism is to disrupt proofreading in bacterial protein synthesis. They can be used alone to treat gram-negative aerobic bacteria, but could also treat gram-positive bacteria if combined with a cell wall synthesis inhibitor, like a beta-lactam antibiotic or with vancomycin. Notable adverse drug reactions include nephrotoxicity, ototoxicity, neuromuscular blockade, nausea, and allergic reaction. Tetraciclines are antimicrobial antibiotics that inhibit bacterial ribosomes, which are the organelles that make proteins. Genes become proteins in two steps: transcription and translation. During transcription, a specific gene on the DNA is read and a copy is made called a messenger RNA, which is like a blueprint with instructions on what protein to build. Translation is also known as protein synthesis, and it's when organelles called ribosomes assemble the protein from amino acids within the cytoplasm. Now, prokaryotic cells, like bacteria, have smaller ribosomes than eukaryotic cells, like those found in humans. Bacterial ribosomes are made up of a 50S subunit and a 30S subunit, which combine to form a 70S ribosome. Eukaryotic ribosomes are made of a 60S and 40S subunit that form an 80S ribosome. Since these proteins are different, we can create medications that selectively interfere with the bacterial ones. In both eukaryotic and prokaryotic cells, protein synthesis involves initiation, elongation, and termination. In bacteria, initiation happens when the 50S and 30S subunits bind to the mRNA sequence to form a ribosome mRNA complex. The mRNA serves as a blueprint for the protein that'll be synthesized. It's made up of 3 nucleotide long sequences, called codons. Transport RNA, or tRNA, carrying different amino acids can bind to these codons with their matching anticodons. The complete ribosome mRNA complex has 3 sites where the tRNA can enter and bind. These are called the A, or amino acyl site, the P, or peptidyl site, and the E, or exit site. Elongation starts when the first tRNA carrying a formal methionine amino acid, enters the P site and binds to the start codon. This causes a conformational change in the ribosome, which unlocks the A site for the next tRNA. A process called proofreading happens here, where only tRNAs with the matching anticodon can bind to the corresponding mRNA codon. After the next tRNA binds at the A site, the amino acid detaches from the tRNA in the P site and gets attached to the amino acid in the A site by the enzyme peptidyltransferase. This step is called transpeptidation, because the peptide chain is transferred from the P site tRNA to the A site tRNA. Now the A site has the newly formed peptide chain dangling from it, while the P site has an empty tRNA with no amino acids. In the final stage of elongation, the ribosomes slide across the mRNA, and the A site sits above a new codon. The tRNA that was in the A site slides over to the P site, and the tRNA in the P site slides over to the E site. Now a new tRNA with a matching anticodon binds, and the process repeats until a long peptide chain, called a protein, is synthesized. Finally, termination happens when the ribosome comes across a termination codon on the mRNA. There are no tRNA anticodons that can bind to these, so they signal the end of protein synthesis. Tetracyclines were originally derived from soil-dwelling Streptomyces bacteria. They get their name from their structure, which involves 4 rings bound together, so tetra for 4, and cyclins for rings. Tetracyclines bind to the A site on the 30S subunit of the ribosome, which inhibits the binding of tRNAs to the mRNA ribosome complex, and shuts down protein synthesis before it even starts. Tetracyclines are divided according to their duration of action. Short-acting tetracyclines, like tetracycline itself, have a half-life of around 8 hours, and long-acting ones like doxycycline and minocycline last for 16 hours or more. Now, some bacteria developed enzymes that can break down tetracyclines, or they have proteins on their membrane that could pump the medication out. So we developed glycyl cyclines like tigacycline, which are modified tetracyclines with an extra dimethylglycolamide side chain. This prevents them from binding to bacterial proteins and enzymes so they can overcome bacterial defense mechanisms. Administration of tetracyclines is usually topical or per oral, but tigacycline is intravenous only. If used per orally, tetracyclines should not be taken with food because they readily bind with magnesium, iron, aluminum, and calcium, and the resulting molecule is then unable to be absorbed. That's why tetracyclines are not given to patients on iron and calcium supplements, as well as antacids, which can contain aluminum salts. Tetracyclines are broad spectrum, meaning they can target a wide variety of gram-positive and negative bacteria. They're often used to treat severe acne or community-acquired pneumonia. Tetracyclines also tend to accumulate within human cells, which makes them useful to treating intracellular pathogens. Common intracellular bacteria include Mycoplasma pneumoniae, which causes atypical or walking pneumonia, and Chlamydia trachomatis, which causes chlamydia. Less common diseases include those transmitted by ticks, like Rocky Mountain spotted fever from Rickettsia species, rabbit fever or tularemia from Francisella tularensis, and Q fever, which is more commonly transmitted by inhaling farm animal fecal particles that contain Coxiella burnettii. For Lyme disease, which is caused by ticks carrying Borrelia burgdorferi, doxycycline is the medication of choice. It can also be given to prevent malaria, which is a parasitic protozoa that infects blood cells and hepatocytes. Both doxycycline and tigacycline are also effective against MRSA, a notoriously tough bug that resists many common antibiotics. For side effects, tetracyclines cause phototoxicity, where patients are much more likely to get sunburned, and tinnitus, or ringing in the ear. They can also accumulate in teeth and bones, which cause permanent discoloration of the teeth and a delay in bone growth. This is particularly important in children under 8 years of age, and in pregnant or breastfeeding women where these medications are contraindicated. Metabolic products of tetracycline are nephrotoxic, and can cause Fanconi syndrome where there's inadequate reabsorption in the proximal renal tubules. Tetracyclines are also not very shelf-stable, and using them after their expiration date can cause liver failure. Other side effects are nonspecific and include stomach upsets, diarrhea, and on rare occasions, allergic reactions. Now let's make a simple and fun mnemonic that'll help you efficiently memorize and retain all these farm facts. Since we're talking about cyclins, let's have a wacky bicycle race. Now, the short-acting medication of this class is tetracycline, so let's have a pair of twins riding a four-wheeled cycle in last place. Up ahead are the long-acting medications, so we have a minotaur on a bike for minocycline, and a biking doctor for doxycycline. Pulling ahead of the others is a tiger for tigacycline, who's riding a heavily modified hang glider bike because it's a glycylcycline, which allows tigacycline to easily glide over bacterial defense mechanisms. For the bugs treated by these medications, let's use the spectators of the race. First, tetracyclines are commonly used to treat acne, so there's a teen with a lot of pimples. His wheelchair-bound grandmother is also there with an oxygen mask to represent community-acquired pneumonia, which is common in nursing homes. Next, we have the intracellular bugs, so let's rope off an area for these guys. There's a young hiker who's also wearing an oxygen mask, since walking pneumonia is common in young adults. He's standing on a giant clam, which represents chlamydia, to get a better view. Next are the tick-transmitted diseases, so let's have a mountain climber with a red polka-dotted shirt for Rocky Mountain spotted fever. He brought his French pet rabbit for rabbit fever, which is caused by Francisella. His girlfriend is wearing a brunette wig for C. brunettii, and a shirt with a giant Q on it to represent Q fever. She's throwing a giant wedge of lime at the doctor, since doxycycline is the first-line treatment for Lyme disease. But the doctor is distracted from the swarms of mosquitoes chasing him, which represent malaria. Finally, the doctor is about to run over a poor Mars rover for MRSA, which is already ran over by the tiger, because both medications are effective against it. For side effects, we can use the referee of the race. He fell asleep in his chair, and his skin is quite sunburned, to represent phototoxicity. In his arms, there's a crying baby, with big yellow teeth and very short arms to help you remember tetracyclines build up in the teeth and bones, and they should not be used by pregnant women or children. There's an electric fan on the sleeping ref's knees for Fanconi syndrome. Since the race is almost over, his alarm clock is ringing, which represents tinnitus. Next to him is his half-eaten lunch, which is a plate of rotting liver that's been sitting in the sun too long. This represents the hepatotoxicity caused by expired tetracyclines. Alright, as a quick recap. Tetracyclines are antibiotics that prevent protein synthesis by binding to the 30S subunit of bacterial ribosomes, and preventing tRNA from binding. They're broad spectrum, and are used for treating many gram-negative and positive bacteria infections. Since they accumulate in our cells, they're very effective against intracellular bacteria like chlamydia. Side effects of tetracyclines include phototoxicity, tinnitus, teeth staining, bone growth delay, nephrotoxicity, and hepatotoxicity. It's contraindicated during pregnancy, breastfeeding, and for children under 8.